Now, in this lecture, we will study how the chromosomes they pair up and make a synaptominimal complex. जैसे हमने पहले लेक्चर में देखा कि किस तरह क्रोमेटिड, क्रोमोसिन और हैप्लॉइड chromosomes they are made. Then the cell it undergoes another process, that is how these chromosomes they pair up, how homologous chromosomes they pair up and make up a complex which is called synaptonomial complex. Now the process which is known as a synapsis, and during this process homologous chromosomes क्या होते हैं? वो क्रोमोसोम जो कि एक तरह के, they are like one and another. And how these homologous chromosomes they are brought together in the site of recombination, जहाँ पे उनको जुड़ना है, जहाँ पे उनको recombine होता है, and they are linked tightly along the entire length by a protein called synaptonymal complex. Remember, जबकि हमने in our earlier lecture we discussed about the cell cycle, we mentioned the proteins. Proteins are very important, and there are a similar proteins are the characteristics protein. खास तौर पे एक तरह की proteins जो हैं वो इसमें काम करती हैं, and they are known as synaptonymal complex proteins, and they bridge the gap between the chromosomes. देखें, अब in the cell the chromosomes they are separated, and there is almost 100 nanometer distance between them. But how they are brought together, how they are complex, that is we will study in this lecture. अब इस कार्टून में देखें, इस तस्वीर में there are two homologous chromosomes. One is indicated by blue color, and the second one is indicated by the red color. And there are centromeres, sister chromatid, homologous chromatid which are there, but they are also joined together. अगर आप देखें कि एक क्रॉस सेक्शन में I have tried to show the synaptonymal complex जो कि उनको जोड़े हुए हैं, जिस तरह के एक तरह की बॉन्ड्स हैं वो उनको जोड़े रखती हैं. Now each homolog is organized around a protein axial core. Remember, again the proteins are very important, and they make some axial core. एक एक्सेस बना देते हैं जिसके इर्द गिर्द वो जुड़ जाते हैं. The synaptonomial complex forms when these homolog axes are linked by the rod-shaped transverse filaments. जिस तरह के मैंने पहले दिखाया हुआ है, rod-shaped transverse filaments, they are there and they are joined by that. The axial core of each homolog also interact with the cohesion complexes. अब आप देखें कि there are also another complexes which are cohesion complexes. Cohesion क्या होती है? Cohesion means आपस में जुड़ने की सलाहियत, आपस में जुड़ने की सलाहियत, जबकि वो आपस में उनको joined together रखते हैं. Just as in this cartoon, if you see the axial cores and there is a cohesion proteins जो कि spring की तरह हैं, and that is the 100 nanometer distance between these two chromosomes, which has been simplified schematic drawing of synaptonomial complex इसमें दिखाया गया है. Now the morphological changes that occur during homolog pairing are the basis of dividing mitotic prophase into 5 sequential phases. Remember, this is very important statement. ये एक बड़ा important असूल है कि the morphological changes उनमें जो आती हैं उसमें during the homolog pair, जब homolog pairing हो जाती है तो वो क्या हैं और किसने. There are 5 sequential stages. 5 बिल तरतीब इसमें stages हैं जिनमें कि ये occur होता है. The one is known as the leptoproteinase zygotine, piketine, diplotin, and diakinesis. And we will study all these 5 sequential stages one by one again. Next. Now, what is during the morphological change of leptotene? During the leptotene, the morphological changes are that the chromosomes, they condense tightly together. लेप्टोटीन में पहली फेज ये हो गई कि जो क्रोमोसोम हैं वो टाइटली एक दूसरे के साथ जुड़ जाते हैं. देन zygotin, a lattice of protein is laid down between the homologous chromosomes in the process of synapses, forming a structure called a synaptonomial complex. जिस तरह के हमने पहले पढ़ा कि एक लैटस. व्हाट इज ए लैटस? लैटस मीन एक प्रोटीन का स्ट्रक्चर that develop that makes them together, to join them together. Then there is a pachytene that begins when the synopsis is completed. जब synopsis जो है ये complete हो जाए तो उसके बाद morphological change is the pachytene, and the synaptonomial complex holds the two replicated, जो के दो replicated chromosomes हैं जो replicate हुए हैं उनको closely together रखती है. Then there is within the synaptonomial complex the DNA duplex, it unwinds. DNA जो है ये एक wound structure है, but during then it unwinds. ये खुल जाता है कहीं कहीं से, and a single stranded DNA form the base pair. As you remember, the DNA is composed of different base pairs, और वो जो base pairs हैं they are open, they are available with complementary strand, and then they join together with the complementary strand. Complementary strand क्या होते हैं? Complementary strands वो हैं where they join with the next उनका जो है strand, उसमें वो join हो जाते हैं and they complex together. The synaptonomial complex thus provide the structural framework that enables the crossover. Crossover ये है कि उनके जो कॉम्प्लीमेंट्री स्ट्रैंड हैं, जो कॉम्प्लीमेंट्री डीएनए हैं, वो वहाँ पे जुड़ जाते हैं और एक नया जो है स्ट्रक्चर वो अमर्ज हो जाता है. उसके बाद डिप्लोटीन है. एट द बिगिनिंग ऑफ डिप्लोटीन द प्रोटीन लैटस ऑफ सिनेप्टोमियल कॉम्प्लेक्स इट डिसेंबल्स. अब पहले असेंबल हो गया, अब ये जो स्टेज आई मॉर्फोलॉजिकल स्टेज, दैट इज द डिप्लोटीन स्टेज इज द डिसअसेंबली. अब उन्होंने अलहदा तो होना है ना, ये जो क्रोमोसोम्स और क्रोमेटिड बन गए हैं. देन द डिप्लोटीन इज ए पीरियड ऑफ इंटेंस सेल ग्रोथ. ड्यूरिंग दिस प्रोसेस, रिमेंबर जब सेल प्रोसेस हो रहा है तो सेल ये ग्रो भी कर रहा है, डीएनए सेपरेट हो रहा है, क्रोमोसोम जुड़ रहे हैं, एंड देयर आर ऑल सॉर्ट ऑफ प्रोसेसेस विच आर वर्किंग टुगेदर. बट इन द डिप्लोटीन स्टेज क्या होता है कि सेल जो है इट ग्रोज, इंटेंस सेल ग्रोथ. इंटेंस सेल ग्रोथ होएगी तो ये सेल जो है ये डॉटर सेल में बनेगा. एंड ड्यूरिंग दिस पीरियड द क्रोमोसोम दे डीकंडेंस. डीकंडेंस का मतलब है पहले वो जुड़ जाते हैं, अब वो अलहदा हो रहे हैं, एंड इट बिकम्स वेरी एक्टिव इन ट्रांसक्रिप्ट. ये ट्रांसक्रिप्शन जो है ये शुरू कर देता है. दैट इज द अनदर the term we have come across, and you know very well about the transcription which the DNA performs. And then there is a diakinesis. That is at the beginning of diakinesis, this morphological stage, इस stage के ऊपर क्या होता है? The transition into metaphase transcription ceases. Metaphase जो है ये बंद हो जाती है, and the chromosomes they recondense. जो क्रोमोसोम जो हैं जो क्रॉस ओवर हुए हैं वो रिकंडेंस हो जाते हैं. जस्ट एज शोन इन दिस डायग्राम you can see the leptotone, that is the homolog pairs, they are there, zygotine, diaptine, and the central element is this, where the one is chromosome is from the maternal and another one is from the external. Now, in this cartoon, we have shown the different process of morphological process of the chromosome division and the chromosome formation. And as you see, there are different stages: leptotin, picotin, diplotin, zygotin. And one is the maternal chromosome, another one is the paternal sister chromatid, which is there. And inside, there are the core. And it is just like a zipper. जिस तरह zipper नहीं होता, उस तरह है how they will separate. And then final stage, that is the chiasmata, and after the complexes have disassembled, the individual crossover events between non-sister chromatid can be seen as interlog connections called chiasmata. Chiasmata is the plural, and the singular is chiasma, which you have come across in your textbook frequently. This plays a crucial part. ये जो स्टेज है, ये एक बड़ा ज़रूरी और आम रुकन है जो कि होल्डिंग द कम्पैक्ट होमोलॉग्स टुगेदर, जो कि होमोलॉगस क्रोमोसोम्स हैं, उनको करीब रखने में करता है अमल. और ये आप देखें, ए बाइवेलेंट विथ three chiasmata resulting from three crossover events. In this case, the one which is the black, that is the original, where there are three crossover, and you can see the red, black, and another shadow one, they are crossovering together in origin.